Can everyone hear me okay? Excellent, great. My name is Shaun. I'm a postdoc with David Jones at UCL, and I'm going to tell you about evaluating AlphaFold for multi-domain protein structure prediction. And multi-domain proteins are kind of important and interesting problem, as many of us know. A very significant fraction of all proteins in eukaryotes and prokaryotes contain more than two domains. But if you look at the content in the PDB, a very small fraction of these chains have more than two domains. And what I've got on the right there is a pie chart of PDB chains in ECOD, which is a database that classifies protein domains. And you can just take that database and look at how many domains are in each PDB chain. And I haven't redundancy reduced this, but it kind of makes the point that it's fewer than around 9% of the chains in ECOD have more than two domains in them. This is a problem. Like there's a significant gap here because many of these proteins are potential drug targets. They're of huge interest to those involved in drug discovery efforts. And now that we live in a post-AlphaFold world, it's kind of interesting to ask, well, are these models that AlphaFold predicts accurate enough if we look at sort of the reference structures that we have in the PDB? And it's worth mentioning also that most systematic evaluations of AlphaFold's accuracy to date have mostly focused on single domain accuracy, and there we know it's extremely good. We have seen a few limited evaluations of multi-domain accuracy as well, including in CASP. But these evaluations don't seem to answer this key question that we're always interested in when we're evaluating a method based on machine learning, which is, can this thing accurately predict things that it hasn't seen during training? The concept of what these things are in this context is a very difficult and interesting one. It's very straightforward to do this for single domain proteins. It's not that easy for multi-domain proteins. But as an initial sort of evaluation, given that there now exists the AlphaFold database, we assess the structures that AlphaFold has produced in this database, and we looked for matching PDB entries that were released after AlphaFold's training set date. I want to stress here that we use the SwissProt version of this database because that was the latest release at the time we did the analysis. There's really no reason you can't extend it to the full database that we have today. So this is the method we used. We find PDBs released after AlphaFold's training date, which is somewhere in 2018. We use MMSeqs to match these sequences to the AlphaFold database sequences. And then we identify a list of hits, and we pick the one that has the highest TM-aligned score to the AlphaFold model. So we're being generous to AlphaFold here and saying, these proteins probably have multiple conformations and stuff like that. And so as long as it's predicted one of them well, we're happy. And then we divide these predictions into three categories, whether it's been seen during training, so where the structure already matches something that we know is in AlphaFold's training set, where it doesn't match something in the training set, but there are templates available. So we look at the list of templates used to make each prediction. And then the third category is where it's neither in the training set nor is there a template available. And we expect the predictive accuracy to vary as a function of these three categories. And sure enough, that's what we see. On the right hand, I have a box plot of the three categories that I showed you. At the bottom is structures that we think AlphaFold will have seen during training. On the x-axis is the TM-aligned score to ground truth, so higher scores are better. A score of 1.0 means you have an absolutely perfect prediction. And as you can see, the median value is very high. It's well in excess of 0.9. In the case where templates are available, even if AlphaFold hasn't seen it during training, the accuracy, again, is very high. There are only 3 instances in this set where no template was used, and there we get this very mixed sort of data set. It's literally a box plot of 3 points. It's, you can't really tell anything from it. But yes, you will have noticed that there are some long sort of tails here of low accuracy, and I looked into some of these structures. They contain things that are where the ground truth structure isn't from an X-ray experiment. There are some unfolded state NMR structures in there, so not really a good ground truth. There are alternative conformations in there. One of these is a calmodulin that's been predicted in just one species in one of the 2 conformers. And then there are obligate multimer interactions, things like domain swap dimers and so on. So those are all very sort of allowable exceptions. So we've seen that template availability is important. And so we want to see, well, okay, what fraction of these models are actually covered by templates? And in the multi-domain context, we're interested in seeing, well, can AlphaFold sort of copy or steal the relative positioning of domains from a set of templates. And you don't have to limit yourself to just the one template there. You can say that if template A covers the first half of the structure and template B covers the second half, you can probably impute the entire thing. So we look at this set of chained templates with the notion that a long chain of templates could possibly be used to predict the entire structure. And so we looked at what stretches of residues are covered by templates, and if they overlap by a certain amount, we kind of merge them and say that's one template. And on the right, you have some box plots of TM-align scores versus the percentage of residues covered by the largest set of such overlapping templates. And you find that there is this correlation. And again, you have that tail of low accuracy, which I spoke about in the previous slide. So yeah, template availability clearly matters. It definitely helps if AlphaFold is using templates. And so one of the things we realized in doing this is that the AlphaFold database is not a great set for evaluating generalization, which was that kind of main sort of question that we're interested in. Most of these are covered in AlphaFold's training set, or they're covered by templates. And it's kind of hard to see where, if any, there is any generalization going on. And so we have to come up with our own data set. And what we do is we start from multi-domain PDB chains released after the training date. And crucially, we decided that if you just exclude chains from your test set that contain one domain in common with anything in the training set, you just have no data left. And so what we decided was that, well, we're interested in the relative positioning of domains. So why don't we look at pairs of domains? And so we said that in our test set, we will retain chains that contain at least one domain not seen in combination with any of the other domains in that chain within any single training example. And the idea is that if AlphaFold has seen those two domains together in a single chain during training, it's probably going to be able to predict that. And so any such domains that are left, we kind of look at what's in the training set, we look at pairs in the training set, and then we mark proteins in our test set according to whether that pair has been seen. And if anything's left over, then we say that's a valid test case. And so we call these regions that haven't been marked by the procedure unseen regions, or regions not in the training set, just to keep the terminology simple. But we're talking here about interactions. Domain definitions are taken from the ECORD database, just as a technical point. And I am making scripts and data available to generate such a data set for any machine learning method. I think the main thing you need to input is the date at which you trained the thing. So what release of the PDB you worked in. So that will be made available. And here's a couple of summary figures of what's in this test set. We have 195 structures, most of them less than 600 residues in length, most of them 2 or 3 domain. Again, that reflects the distribution that we know from the PDB. And yeah, there are some longer sort of cases in this set, which makes things interesting. So what does full length and per domain accuracy look like? On the left here, I have full length RMSD to the native for the AlphaFold2 prediction. So this is a bog standard run of AlphaFold with all the defaults. And you can see that there are plenty of proteins in this set where AlphaFold does really well. We're talking about full chain RMSDs, less than 5 angstroms to the native. Really good predictions. The dependence on sequence length is very low, if anything, which is also good. But as you can see, there are quite a few cases where that accuracy is really, really poor. And those are the ones we're more interested in, I guess. And looking at per domains, as I said, individual domains predicted very, very accurately. So a very large fraction of these within 4 or 5 angstroms to the native, very accurate predictions. There is a long tail that corresponds to things where domains aren't globular in the ground truth as well. So quite a few interesting things here. And so we want to use this information about what domain pairs have been seen during training while doing the evaluations. And so the question is, of course, how frequently or how much are these domains placed incorrectly if they haven't been seen in combination with anything else in the chain during training? And to do this, we used a program called Profit from Andrew Martin's group, which allows us to do these partial superpositions. And what we do is we align the prediction and the native structure on the domains that we think AlphaFold will have seen during training. And then we just measure the deviation, keeping that superposition constant. We measure that deviation on the unseen regions. And this gives you an idea of like how much error in the prediction is due to or is correlated with the fact that these domains have been seen together. And this is what it looks like. So on the x-axis, we have the RMSD to the native for regions that we think AlphaFold has seen in the training set. So this is the parts of the chain that we actually align. And on the y-axis, we have the RMSD of the regions, keeping that superposition fixed of the regions that we think AlphaFold won't have seen in combination. And so many of the points are above this line, as you might expect. So we're saying that on average, the error on those parts of the chain that haven't been seen is greater than the error on the parts that have been seen. So we think AlphaFold clearly has problems placing domain pairs that haven't been seen during training. But what's really interesting on this plot, and there are several things interesting about this plot, but one of the interesting things, if you just look on the x-axis, both the axes are on log scales, by the way, because some of these deviations are very large. Even if you look at just the parts of the chain that have been seen during training, there are examples where AlphaFold hasn't done very well. Not entirely sure what the reason for this is, but here's one example. The native structure is in green and the predictions in blue. And you can see that the entire chain is kind of collapsed. And to be fair, the native structure here is a heterodimer, is a dimer of heterodimers, and they're kind of extended in conformation like that. So that could be a reason, but as we know, AlphaFold doesn't know physics, so we've seen examples where it can do that, where it can predict these structures as if the other partner is present, but it hasn't done that in this case. And we have a mixture of X-ray and non-X-ray structures, and you can see that there is a little bit of a dependence on this measure, depending on whether your ground truth is an X-ray structure or not. Encouragingly, though, 41 out of 76 targets have an RMSD of 2 angstrom or less on regions seen during training. So just drawing a sort of vertical line here, that's saying that these parts really align quite well. And there are actually targets within this kind of 5 angstrom, 5 angstrom bracket where we say overall the chain is predicted pretty well. But yeah, there are these deviations, and on average, there is a tendency for it to not place these domains correctly if they haven't been seen. One limitation of this analysis is that it can only be applied to chains that have at least one domain seen during training and one not seen. And for all others where nothing in the chain's been seen together, we have to consider full chain RMSD. And this is what you get for those targets. Many of them predicted very well indeed. So RMSD is under 5 angstrom again across the entire chain. Interestingly, most of the proteins in this set are composed of 2 domains. And we think that that has something to do with it. We also did a kind of sanity check to make sure that we weren't just hitting something that was actually in the training set that we thought for some reason isn't. And so we took these regions and aligned them to the training set using TM-Align, and we didn't find any strong hits. So this appears to be genuine sort of evidence for generalization. And also it's worth mentioning here that AlphaFold2 won't have seen many 3 or more domain training examples just because there aren't that many in the PDB. So knowing all of this, we wanted to go a bit further and say, well, can we kind of coax AlphaFold to produce more accurate models for difficult targets in this set? We tried a few different things and, increasing the number of recycles and whatnot. And we settled on using this method called AF-sample from Björn Wallner, which did well in CAST 15. It was designed originally for multivariate predictions, but we reasoned that, with the kind of aggressive sampling that it does, it might work on this problem as well. The way it works is it turns on a few switches in AlphaFold that turned off by default. So it enables this thing called dropout, which just leaves parts of the network out in the forward pass. You can enable or disable templates, and you can enable additional recycling. And because we're working with single chain predictions, we included the monomer PTM preset with and without templates, and we sampled a total of 10,000 models per target. This is incredibly computationally expensive, and I've done it so you don't have to. So this is what we get. On these 35 cases, AF-sample generates at least one model with a TM score improvement of at least 0.1 units in 15 out of 35 cases. 9 of these achieve a TM score of 0.9 or greater, indicating that the model is very, very good, it's highly improved. As I said, the downside is that this method is computationally very expensive. Running AlphaFold in the forward pass on a large protein 10,000 times is not for the faint of heart. It's not the most efficient method in the world. And somewhat disappointingly, these improved models are not generated very frequently. So on this plot, we've got the best TM score achieved by the default AlphaFold and the best TM score achieved by AF-sample. And this is assuming that your model selection is perfect, right? So you've got this pool of 10,000 models and you can pick out the best one. This is what you get. And so here's some examples of the improved models. Again, the native structures in green and the predictions in blue. And you can see that AF sample does nail it in some of these cases, positions one of these domains into the correct sort of orientation. Model selection is a problem with this protocol. As I said, these models are not generated very frequently, and very annoyingly, they're not always identifiable based on PLDDT, PTM, or PAE. So what I've got here on the right is the TM score loss when selecting by PLDDT. So how much of a TM score improvement do you miss out on if you select your best model based on the highest PLDDT in that set? And I've marked in red here some targets where that missed model had a TM score greater than 0.9. So this is like an almost perfect prediction. And you can see that that loss is positive in most cases. In other words, you're almost always missing out on a good model. In most cases, that loss is actually quite small and you can probably tolerate it. But the best cases where the best models have been generated, you can't identify them based on PLDDT, PTM, and PAE looks the same. These measures are highly correlated. They are simply a measure of AlphaFold's confidence in the prediction. And I've actually looked at the PAE plots for some of these, and very disappointingly, the PAE for the highest TM score model looks worse than for the highest PLDDT model, which is very annoying. And I've also tried standard decoy clustering methods, and again, they just identify the top models as outliers in many cases. And again, that's just because of how these clustering methods work. So there's some work to be done here in terms of how we improve the quality of these models. So to conclude, we've developed this curated test set to evaluate multi-domain predictions that's based on domain pairs not seen during training. AlphaFold2 exhibits reasonable generalization to unseen domain combinations, but that drops off for longer targets and those with more than 2 domains. More accurate models can be obtained using aggressive sampling strategies, but these models are generated infrequently. They can't always be identified, and this is a terribly inefficient way of doing it, particularly if you want to update the AlphaFold database. It's just not practical. So there's a need here for some new thinking and new methods, and I'm happy to see that other groups have been doing that as well. I know Yang Zhang and a couple of others have also developed methods to improve these predictions, but ultimately, the key question is, can they do things beyond what they've seen in training? And data sets like this might help in that regard. So with that, I'll conclude and just thank members of the UCL Bioinformatics group. My colleague Andy helped me with some of the figures for this talk, and BBSRC for funding. Thank you very much.
Are there any questions for Shaun? Can you come over? We didn't solve the problem of the microphone yet.
Yeah, hi, thanks for the great talk. Do you find that the PLDT scores of the bridging section between domains are worse when AlphaFold has misplaced a domain than when it's correctly placed it?
Yes, I mean, they are. Yes, that does appear to be the case in general. The thing I was hoping is that when it has placed them correctly, that improves. It doesn't.
Oh, okay, so you can't differentiate based on the PLDT
No,
scores of those bridges?
no,
Okay,
that's
thank
it.
you.
Hi, very nice work. So when I think of multi-domain proteins, I think that for many of them, there won't be a good reference structure that you can say that the 2 domains, relatively speaking, are in this orientation, because based on the linker's flexibility, there's no good reference structure for many of them. So how do you justify using a reference to quantify things?
This speaks to a number of issues, but the short version of that answer is we limit ourselves to things that are in the PDB where the full length structure is available. To go into it a bit more deeply, if you ask 5 different people who are end users of these models what they want, they will give you 5 different answers. These things are conformationally flexible. That's just life. In the context where we're trying to evaluate machine learning methods, we have to have some kind of ground truth. And really, at this point, this is the best we have. So that's it, I'm afraid.
So first of all, thanks a lot for your talk. It was really great. You have divided the PDB samples into structures that are obtained from crystallography and others. Have you also further subdivided the other structures into, for example, cryo-EM and NMR structures?
Yeah, somewhat. I've looked into this a little bit. So NMRs tend to be okay, but what I have seen in that data set is that there are some where the structure is meant to be an unfolded reference state. So that's really not a good ground truth to evaluate on. That's why we see some of those low accuracy structures. But yes, they tend to be better than cryo-EM in general.
Okay, and would you say then that this is an issue that AlphaFold has learned things from structures that are actually only, or maybe only present in a crystalline way?
Yeah, I mean, we know from their paper that that's something they actually explicitly specify. Yes, I mean, there may be some bias in that sense. It was never designed to predict conformational heterogeneity, flexibility, this type of thing. So I guess, again, it's one of those things you have to choose your ground truths very carefully. And there, I would say that it probably makes sense to do that, and you can take the resolution of the structure into account. But I think that's about the best you can do.
Yeah, cool. Thanks a lot.